Hello Year 11, right, this is for separate science students only. If you are a trilogy science student, so at Horizon, if you are in set 2 or below, then you do not need to watch this video. Right, 11A1 and 11E1, you were sent some exam questions on email, so I hope you've had a look at them. We're going to go through some of them. There were quite a few topics that didn't come up on a separate paper. I've put them in this box here. There's absolutely no way, we're not going to get through these in about half an hour, so I'll go through as much as I can, but you have got all these questions, I've emailed them to you, okay? So I've spent all day cutting and sticking and making sure that I've got all the pictures that I wanted, because I'm not drawing them all, I ain't got time. Right. Firstly, the eye. The eye did come up. I need some blue tack because it's gone all curly because of my bad cutting and sticking. The eye did come up last year, but it didn't really go into much detail. It definitely didn't talk to you about accommodation in the eye. So if we have a look, we're firstly going to look at responding to light levels. So when you go into a dark room, your pupil will dilate. So in dim light, your pupil will dilate. In bright light, your pupil will constrict. And there are sets of muscles that you need to know about here. And the easiest way to remember it, and believe me, this is a topic that I always forget. I only teach it once a year and it takes a lot to go back into my head. You've got two sets of muscles you need to worry about. You've got the circular muscles and you've got the radial muscles. Remember, C. When your pupils constrict, it is the circular muscles that contract. Muscles work in antagonistic pairs, so that if your circular muscles are contracting, your radial relax. So if we look at the dim light, if we flip that round, so your circular relax and your radial contract. And that is a response to change in light levels, okay? Where a lot of people slipped up last time was what has caused the change between two pupils, and you put light. It is the brightness of the light or the dimness of the light. The next section didn't come up last year and it's very, very difficult. It's come over from physics. It's called accommodation. I'm going to spell this wrong, apologies. Accommodation in the eye. So basically the focusing on different objects. When you focus, or when your eye focuses, we need to make sure that the light rays meet at the back of your eye, which is the retina. Okay? If you are focusing on a distant object, we need to make sure that our lens is thin. If we are focusing on a close object, we need to make sure that our lens is thick. Okay? And in this one, it's the absolute opposite of what you would think. You've got muscles and you've got ligaments, okay? So with this, if you want your lens to be thin, then, probably going to get this wrong, tell me if I do miss, because I'm feeling a bit under pressure now. We've got the muscles and we've got the ligaments. So the ligaments, to make your lens thin, will tighten. Yep, so the ligaments tighten. And in my brain, what I want to say is what would cause that would be the ciliary muscles to contract, but that is the absolute opposite. Your ciliary muscles relax, which makes your ligaments tighten. And I can never spell this word, so ciliary muscles relax, the ligaments tighten, which is what I would think to be the opposite way around. So that's why it always confuses me. In this one, if we're focusing on a close object, your ciliary muscles contract, the ligaments loosen, and this allows your lens to become thicker. Okay, so that is the first bit of the eye. Not got anything on the back of that page. We've also got correction of eye problems. So I'm not going to get a chance to do all these exam questions, guys. I'll just go through the content of it. Okay, so if we have a look at these two pictures, what we've got two problems with eyesight. So in the first one, the light rays are focusing in front of the retina, so they're falling short of the retina. This is called myopia. Okay, on the second option, or the second problem, the light rays are focusing beyond the retina, and this is hyperopia. The correction of these vision problems, if you are myopic or have myopia, you would need to use a concave lens. If you have hyperopia, you would need to use a convex lens. These can also be called converging, diverging lenses. Okay, you have got this exam question here, if you checked your emails, and it's basically saying what is going on from A to B and to explain how the changes occur. So quickly, if we go from A to B, what has happened? The pupil has dilated. If we remember, if it's dilated, it must be in dim light. If it is constricted, it must be in bright light. And to think about it, remember the CCC. So if the circular muscles have contracted, here the circular must have relaxed. And so the radial must have contracted. And that will get you 4 marks. So you'd say what the change was, you'd say what caused it, so the dim light, and you'd say what the muscles were doing. Okay? Another part that didn't really come up, guys, was the brain. So I know you'd all, well, everyone last year had revised that in quite a lot of detail and didn't even come up. So the brain is new to our specification. It was new. So if you have a look, we'll ignore this exam question for now because we've just been through it. If you want to have a go at it on your own, you can. Okay. So the parts of the brain. We've got this part, which is the cerebellum. The cerebellum is involved in movement and coordination. We've got the medulla. Full name medulla oblongata, you can just call it the medulla. This is involved in basal metabolism. That is also your heart rate, your breathing. And the other part that you need to know for this part of the spec is your cerebral cortex. You can also call it the cerebrum. This is involved in memory, language, etc. Although it's not on this part of the spec, you could be asked about what this part is in the middle. So this is more spec 5. You've got the pituitary gland, which is part of or near the hypothalamus. Okay, they're the parts of the brain. How we know what the parts of the brain did, we used to study brain damage. So if someone took a blow to the head and they started speaking a different language, then we would assume that a different part of the brain has been damaged than if they lost coordination. So exam questions that you could get: a woman's got a head injury, the symptoms, she can't name things, she can't remember recent events. Let's have a think, which part of the brain could be damaged? It would be the cerebral cortex, or you can call it the cerebrum. A man has a head injury, he starts to stagger and sway. If we consult our little diagram, it would be the cerebellum, because that is involved in movement. Okay, so that's the eye and the brain. That's a very, very short tour. Thank you, Carl Linnaeus, for your expertise on the refraction of the light in there. I've read the comments, very good. So we're going to now look at the kidney. The kidney did not come up It's a very big part of this topic. So we're going to look at the kidney and what it actually does. Then we're going to look at the formation of urea. Okay, so if we have a look, we've got a diagram and we've got some exam questions. Okay, so if we have a look, we've got the kidney, we've got the blood that is entering. The first stage of urine production, the kidney will filter, can't spell, the blood. Large molecules such as blood cells and proteins cannot pass through the filter, so they cannot diffuse into the kidney. So red blood cells and protein stay in the blood. Small molecules such as glucose, urea, and ions enter the kidney. So the first stage is filtration. In the second stage, the kidney reabsorbs useful substances back into the bloodstream. So the second phase is reabsorption. And you need to be extremely specific with your words. The kidney reabsorbs into the blood absolutely all of the glucose. It reabsorbs some ions and some water, and that is as needed. Okay? What we've got left is the urea and some of the water. So in here at the minute, we've got urea, water, and some ions, because we've only reabsorbed as much as we needed. The urea then flows out as dissolved in urine, and it goes to the bladder where it is stored. So they're the stages of filtration, reabsorption, and urine production in the kidney. Exam question wise, describe how a healthy kidney produces urine. So if we're doing it like we should hug our question. So highlight, underline. Was a 5-mark question, you should glance at it, I've cut it off. So I'm not going to write it all out again. I'm going to go through what is the important part. So firstly, the kidney filters the blood. Small ions and small molecules such as glucose, urea, and ions will enter the actual kidney. In the second stage, the bloodstream reabsorbs all of the glucose, because we don't want to get rid of that because it's very useful. So all of the glucose is reabsorbed back into the bloodstream, some of the ions and some of the water as needed. So the ions, water, and glucose are back into the blood to be used. What we've got left in the kidney, we've got the urea, water, and ions, and these leave the kidney as urine to be stored in the bladder until they are needed. Okay? This stuff, urea, though, you need to know where it comes from, and it comes from the deamination of amino acids. Deamination didn't come up last year. We need to know the process of it. So in your diet, you eat protein. You might have people, or you might even go to the gym yourself and have all these protein shakes. A lot of the time, if you don't actually need that protein, it's an absolute waste, because eventually the amino acids that form that protein will be excreted in your urine. So your protein is broken down by protease, you should know this, into amino acids. Okay? This amino group isn't a very pleasant one. It can be toxic to us, so we need to get rid of that. You can't store amino acids. There's no way to store them. They're either broken down into urea, or you can transaminate them into other types sometimes. So your amino acids are broken down first into ammonia, which is highly toxic. That can't stay around for very long. This is then turned into a slightly less, but still quite toxic compound called urea. Urea is then released in urine. The bit where people get mixed up here is that this deamination part takes place in the liver. Urine is made, or urea is removed in the kidney from the blood, and then the actual urine is stored in the bladder. So it involves, or getting rid of this stuff involves 3 organs. Okay? Right. Other part of this, to do with homeostasis. You need to know a little bit or quite a lot about ADH. I'm not going to write anything down for this, guys, but please, please, please look at this diagram. You have had it before, set 1. Okay. So the water content of your blood is controlled by a hormone called ADH, or antidiuretic hormone. This whole cycle is a negative feedback cycle because any change your body puts into action, mechanisms to bring it back to normal. Once it's back to normal, the mechanism shuts off. So if you have a look at this little diagram, you've got normal water content. If you drink too much water, the water content of your blood will be too high. And you've got things called osmoreceptors, and these detect changes in water levels. If the water content of your blood is too high, your brain will make less ADH. ADH stands for antidiuretic hormone. A diuretic is something that will make you wee. So if it's making less ADH, then you're going to be making less antidiuretic hormone. So what happens is, less water is reabsorbed into the bloodstream because your kidney tubules, or the membranes in your kidney, become less permeable to water. This means that more water stays in your kidney and therefore is drained into your bladder as urine. So you wee out lots of very dilute urine and it brings it back to normal. Once it is back to normal, then the mechanism shuts off, otherwise you will become dehydrated. On the other hand, if the water content of your blood is too low, or you've eaten too much salt, then this is noticed again by osmoreceptors in your brain, and your brain will make more ADH. This means that your kidney will reabsorb more water back into the bloodstream, so the water that's left in your kidney will be quite yellow, smelly and concentrated, and you'll not make very much of it, and this brings your whole body level of water back down to normal. Okay? Right. We've got about 10 minutes left if we're rushing. So I'm going to skip over. You've got questions, guys, on kidney treatment. So if you have chance before tomorrow, can you have a look at those? We're not going to get a chance to do it now because I want to jump and I want to look at protein synthesis, definitely. So what we've got, the structure of DNA, you should really know. So it's a double helix made of 2 strands. It's made of a polymer. These are twisted into a double helix and there are 4 bases, A, T, G and C. So if we have a look, we've got our double helix, we've got our bases. A will always pair with T, G will always pair with C. You could be given 1/2 of DNA and you could be asked to write what the complementary side would be. Absolutely fine. So if we look at the exam question, a molecule of DNA contains 4 different bases, W, X, Y and Z. The 4 bases are arranged in a long chain. The chain of bases controls the synthesis of a protein. Remember, proteins are made of amino acids. And your DNA codes for amino acids. The diagram shows a small section of a DNA molecule. This section is responsible for synthesizing a protein for blue eyes. What word is used to describe a small section of DNA molecule that controls the synthesis of a protein? A small section of DNA is a gene. In the cell where our protein is synthesized, remember, we've got transcription, translation, where the protein is actually synthesized, so that would be at the ribosome. Then, gives you a massive diagram of a section of DNA going WXZ amino acid J. This is telling you a lot of information. It's telling you firstly that 3 bases, you already know this, but 3 bases would make 1 amino acid. Okay? And it also tells you that if you've got a different order of bases, you will get a different amino acid. And we know that. Don't think that what they're giving you in an exam question isn't useful. So if we have a look at the question, this is a protein synthesis question. Describe how the protein for blue eye colour is synthesized. How is protein synthesized? This is only a 4-mark question, even though I have cut it down. You don't need to go into a lot of detail about transcription and translation, but we will just look at it now. So, to synthesize a protein, I'm just going to break it down. In the nucleus, you've got your DNA. your DNA does not leave a nucleus. It is too fragile. There are too many things that might harm the DNA out of a nucleus. So in the nucleus, your DNA unwinds and unzips, and then you get a molecule of mRNA that is complementary to the DNA strand. If we remember, in RNA, we don't have a T, we have a U. So it's a U, not a T. So for example, if your chain was, I'll do it here, if your DNA chain was A, G, A, C, I would want to say that was a T for the mRNA strand, but it isn't, it is a U. So it would be U, C, U, A. And that would be my mRNA strand, okay? Once I've synthesized, or once your body has synthesized this mRNA strand, it leaves the nucleus through a pore. This stage, as we know, is called transcription. And your mRNA strand moves to a ribosome, quite a big ribosome, which is in the cytoplasm. Okay? At the ribosome, your mRNA strand is read in threes. It's read in triplets. Okay, so remember that term. Every 3 mRNA bases will code for 1 amino acid. We'll draw a square. So the RNA that brings the amino acids to the ribosome is called tRNA, or transfer RNA. Okay? And this is translation. So every 3 mRNA bases, you get a different amino acid being brought to the ribosome. This will eventually fold over and it will form the protein. Okay? That is a type of information you would need for a 6-mark question. For this one on protein synthesis, you basically get your marks for saying that the bases in DNA code for amino acids. It is read in triplet. The triplet would move to the ribosome where the protein would be synthesized. Okay? Next question. Mistakes sometimes occur when DNA molecules are copied during cell division. Suppose that one of the W bases shown on the diagram was substituted by an X base. What would happen to the structure of a protein synthesized? If you change just one amino acid, you would get a different protein being made. The reason is you would get a different amino acid coded for. Okay? What might be the impact if we were looking at this particular protein? Well, if we think back to the question, it was for eye colour. So if you get a different protein being made, potentially you might get a different eye colour. Okay. Right, that is a difficult topic, guys. If you're struggling with protein synthesis, I would suggest that we watch maybe a bit of free science lessons. Shout out to him. What's his name? Ladies, can't remember. I feel really bad now, I should know his name. But please, feel free to watch free science lessons on that. We have got 5 minutes left. I'm going to make a punt because we've just got so much. We're going to go for cloning. That's what we're going to pick. So, cloning hasn't come up yet. I'm going to show you it in different ways. So, adult cell cloning then is how we can make copies or genetic copies or clones of specific animals that with Dolly the sheep, I think we were just trying to see if we could do it. But you can make specific clones of animals that potentially might be quite high value. You can also use a technique called embryo transfer, where once you've got your embryo, you can make it split or divide many, many times. And then you can have lots of prize-winning animals. So it's usually a sheep, although I've got you another option. So we've got 2 sheep. We've got this sheep here. We've got this sheep here. Okay? So we're going to call it sheep A and sheep B. If we look at this process, we would end up with a clone of one of these types of sheep. So what happens is you would take an egg cell from one sheep and you would remove and destroy the nucleus. From the second sheep, you would take a skin cell or an udder cell, depends what you want to go with, a body cell, and you would take out the nucleus, but you would keep it. Okay, you would then fuse the nucleus and the empty egg, empty egg cell. You give it a bit of an electric shock to make it divide. That's a bit misleading, this diagram. You let it form into an embryo, and then you place it in the uterus of a surrogate. Because the genetic information has come from sheep B, you would end up with a clone of sheep B. To complete embryo transfer, instead of just having one embryo, you could then split that into as many as you wanted and put them into surrogates. The other type of cloning you need to know about is taking cuttings, but that's relatively easy. You would take a cutting from a plant like a gardener would, dip it in rooting powder and then plant it. Tissue culture, again, something that we would do in plants. This is more done on a large agriculture or horticultural, scale. Last question that we're going to squeeze in. I love a horse. I love a picture of a horse. I haven't come up for many years. A horse, surprisingly, is a zebra and a horse. It's produced by sexual reproduction. What is sexual reproduction? I think I've typed this quite a few times tonight. Someone was very interested in knowing what it is. It involves the fusion of gametes. It has characteristics of a zebra and a horse. Why? Because there has been mixing of the DNA. There is half from the horse and half from the zebra. Okay, nice and easy. Then, very similar to the diagram I've just shown you, we've got a picture of a horse and a picture of a normal horse. So this is saying that horses are not able to breed. This would tell you then that horses and zebras are not of the same species. The scientists could produce more horses from this horse by adult cell cloning. The diagram shows how the scientists might clone a horse. So, we take an egg cell from a horse, get rid of this nucleus, it's no use to us. Remove a skin cell from the horse, take the nucleus out. So it will have the horse genes. Put it into this empty egg. Give it a little electric shock. And this cell will divide to form an embryo. This embryo is then placed in the uterus of a surrogate. And when the baby is born, it is a genetic copy or clone of the horse, because that is where the DNA came from. Okay? Shall we do one more? One more question. And I'm going to pick Darwin and Lamarck. So, last one. The rest of the ones we're not going to get to go through, guys, are the things about food production, farming, biotechnology. I think if we were going to guess any, I think potentially making corn or mycoprotein might come up. That's relatively simple though, so you just need to look over the process. So this one, again, natural selection didn't come up very much, speciation did, but natural selection and comparing different theories didn't. So if you have a look, we've got some lovely flamingos. Apologies, it's in black and white. Flamingos feed on organisms that live in mud at the bottom of lakes. Leopards prey on flamingos. Flamingos find it difficult to fly if their feathers get wet. Flamingos have evolved very long legs. How would each of the following theories explain the evolution of these long legs? So we've got long legs. Darwin's theory. As we know from the last video that we made, Darwin would say that there was a random mutation which gave variation in leg length. The ones with longer legs might not have got wet feathers. You could have any sensible suggestion, or you could say they might have been able to go deeper to find food in the water, so they had more food. I'm going to go with longer legs, didn't get feathers wet. If it didn't get wet feathers, this meant that they could escape from leopards. It gives you all this in the question, guys, so we don't need to worry about finding or knowing everything about any type of animal. These then bred and they passed on their genes. That is natural selection through random mutation. So that's Darwin's theory. Lamarck's theory was different. So Lamarck's theory is acquired characteristics. So what Lamarck would have said would be that over the flamingo's lifetime, they stretched their legs to avoid wet feathers. And this was passed on to offspring when they're bred. So the main differences, or the main difference between the two theories, Darwin's theory relies on random mutation. So there's a random mutation, some were better suited than others. The more suited survive, breed and pass on their genes. Lamarck thought that over an organism's lifetime it did something, so a giraffe might have stretched its neck, or a flamingo stretched its legs, and then this was passed on to their offspring, okay? They're the main parts. We can't go through every single bit of this. Before I go though, I'm just going to put these questions under, so if you want to, when you replay it back, if you didn't print my questions off, if you want to have a go at them. So, a little bit on decay. Decay did come up last time. The decay required practical is very similar to the enzymes required practical. Just need to be aware that we can't effectively in lesson demonstrate decay because it happens over such a long period of time. So there's a question there if you want to complete it about the decay of potatoes. Leave it there for a second. We've also got a question about farming and efficiency. Remember, if you're working out efficiency, it's a useful divided by the total times 100. And intensive farming is more energy efficient because we restrict the movement of animals and we keep them inside so that they don't have to maintain their body temperature and they don't use as much energy or need as much energy for movement. Fishing, a little bit on fishing, net size. We need to make sure fishing is sustainable, so having holes in nets will allow, or larger, holes in nets that allow small fish through will make sure that they can breed. And finally, my voice is going, mycoprotein or corn production. You need to understand how a fermentation vat works. Please do not confuse this with the generation of biogas. This is definitely an aerobic setup. The generation of biogas is anaerobic. So this uses a fungus called Fusarium. We pump in sterile air and sterile water and sterile glucose because we don't want any competition with anything else for the fungus. Then we make corn out of it. Okay? Right. Please watch these videos back. All right? Have a go at the exam questions. We will be around tomorrow for your boosters. I'll be around before school if anyone wants to come in, but you have got your English exam, which is kind of important. But anyway, biology I would say is more so. Right, I better go before I get myself in trouble. Good night, Year 11. Thank you for watching, and thank you to Mrs. Froggy and Miss Hughes for giving up their time. Thank you.